Right. Good morning, everyone. Thank you for joining. It's a great pleasure to have today Allison Barnes and Jonathan Ojeda, that they are going to be talking about this large multilateral project where several groups are involved, and mainly showing the goals of the project, and a little bit detailing a review article that we're all writing together, and that will be coming out in the future. So, Jonathan, Allison, please take it away.
Okay. Well, hi everyone. Yeah, we're like Raven said, we're just gonna talk to you about this perspective paper that explains what the CERCA project is all about. And I would like to start talking about uh the agricultural nitrogen cycle and how we need a second nitrogen revolution. So we have experienced one nitrogen revolution in agriculture. Yeah, so we can think of the nitrogen 0.0 as a crop rotation and manure systems that were developed and enabled more productive agriculture. But those systems, they required massive labor and land inputs, and many people still faced food insecurity. Then we had the invention of the Haber-Bosch process, which enabled the massive production of synthetic fertilizer, and then productivity gains and increased a lot land use efficiency. 50 to 60% of the nitrogen in our bodies today comes from these process. So this revolution significantly reduced global food insecurity. Um, and then a project that it has been working in, and probably he can tell us about this some other time, is this new revolution about the nitrogen cycle, which is about increasing the efficiency of this nitrogen to 1.0 and making it nitrogen 2.0, which is more focused on synthetic nitrogen and the circularity of the whole system to recycle and reuse that nitrogen throughout the agricultural production system. But yeah, the problem of the current nitrogen fertilization is that it improves productivity, but at a cost. If we can think of the input of nitrogen and then how it goes through the agriculture and the food production system, and there's a lot of leakage of nitrogen along the way. Croplands are like 50%, 55% efficient, and then livestock is only 23% efficient. And all of these leaks lead to a lot of money lost, a lot of water pollution, and greenhouse gas emissions. So yeah, it is not an efficient system. And I think we think mostly it's a matter of scale. US maize cultivation is around 36.4 million hectares, so it's a lot when you think about this inefficiencies. And then nitrogen fertilizers contributes to greenhouse gas emissions through nitrous oxide emissions that result from soil microbial activities like nitrification and denitrification in spring before the crops are established. Then the production of the actual synthetic fertilizers represents 10% of agriculture-derived greenhouse gas emissions. And then the nitrous oxide emissions from corn soybean rotations in the US are actually 5 times higher than the global average for agricultural soils. Leach nitrogen from these agricultural production systems pollutes water. 70% of the nitrogen and phosphorus delivered to the Gulf come from agricultural processes in the US. Corn and soybean rotations account for 52% of that nitrogen input. There's data that suggests there's a linear relationship between the increased nitrogen fertilizer application and higher nitrogen concentrations in US watersheds. Um, there's like a example of the dead zone of the Gulf, which is the size of New Jersey State. So yeah, it's a big problem the nitrogen that leaches to water bodies. And then the nitrogen fertilizers represents 20% of the farmer's budget, and it is highly volatile. So there is a lot of reliance in the nitrogen fertilizer. Um, so yeah, um, there's a there's a problem with the nitrogen, but we still think maize is the right crop to be working in. Mainly because the nitrogen use efficiency of maize is stands around 64% and has increased a lot throughout the last century. It is higher than other crops. Wheat, it's at 45% in the US and rice is at 41%. Advancements in breeding, genetics, and management have significantly boosted maize nitrogen use since the mid-70s. And maize is land use efficient. Maize yields in the US have increased by 550% since the 1920s, while the land area dedicated to maize cultivation has remained practically the same. So that is a big success. And maize is still a great crop. I'm pretty sure most of you have probably seen this um, plot of yield gains across the past century. So yeah, maize is the right system to go with. So how do we improve this nitrogen efficiency? Um, I think we first have to focus on the fact that maize has two key anomalies. It has a massive seed with lots of nitrogen per seed, which wouldn't be a bad thing. But then the protein that is in maize seeds, it is not digestible. So herbivory pressures during grass evolution resulted in the production of or the accumulation of 60% of plus prolamines in seeds, which have low lysine content and low digestibility. Then 10,000 years of domestication just resulted in an increase of the protein per seed, and that yeah happened with the increase in seed size. And then in the last 100 years, we've got to a number of 1.2 trillion kilograms of grain produced. So if we think about the maize protein content is around 8 to 12%, and we multiply that, it is a lot of protein that is being allocated to this grain production. And if we think that the seeds are 60% of that protein and there are bad quality proteins, then we have a problem because they're the main nitrogen sink, and that nitrogen is not being used in downstream processes, mainly because corn is mostly cultivated because of its starch content, not really because of its protein content. All of the downstream processes are interested in the carbohydrate. Um, the another anomaly is that maize is a tropical species grown in a temperate environment. So if we think of the origin of maize, there's like 3 times the growing degree days that we have in the U.S. Midwest, where corn is cultivated extensively. So that is an anomaly, which results in maize in the maize nitrogen cycle being out of sync in temperate environments. So if we take a look at the soil nitrification rate, which I'm presenting here in this plot, we can see how it starts early in the spring when maize is not yet there in the field. So we have a lot of nitrate that is being produced here by the microbial processes in the soil, and that is not being used. Um, and this is because of the cold temperatures that happen through early April that don't allow maize to be planted earlier. We typically have maize planted in the Midwest like around the 1st of May, probably mid-April, mid-May. And then we have this nitrogen. If we actually plot the plant nitrogen uptake, we can see how these two don't match. There's an asynchrony, which leads to nitrate leaching and nitrous oxide release. Um, And then there's actually a lot of removal of the nitrogen that is being applied to the field, to the grain in these proteins that are not used in downstream processes, like I mentioned before. And there's also not a lot of crop residue nitrogen returned to the soil because all of that nitrogen is being removed in the seed. So yeah, we think of CERCA as a solution to this, and then I'm gonna hand over the microphone to Allison to continue the presentation.
Yeah, so the way that we think that we can improve this is thinking about CERCA maize nitrogen cycle. And so the way that this works would basically be to extend a nitrogen uptake window, which in turn also extends the light interception window and increases yield, and also takes advantage of the spring precipitation window as well. And so the first way that we can think about putting these two curves in synchrony is to push our planting date earlier. So our planting goal here would be April 1st. And what that means is that we need to improve cold tolerance in maize so that we can have that planting date even just 2 weeks earlier, to fight off any of the potential late freezes. The animation, Jonathan. Thank you. So the first thing, yes, is cold and frost tolerance. And so that will allow corn to grow earlier during the beginning stages of the nitrate leaching window. Okay. And then another goal that we have is to decrease the amount of nitrogen that's remobilizing to the grain. Because grain is not being used as a protein source, we think that we can decrease the amount of nitrogen that's actually going into the kernel and instead push it into the cobs or roots, where it can then stay in the farm system and not be removed from the field and need to be reapplied. And so that will involve remobilizing nitrogen to the roots after the black layer has formed, increasing the crop residue nitrogen return, and then also looking at BNI stover that can be left over the winter and into the spring until you reach the next planting. So overall, the goals are to improve seedling cold tolerance and to recycle and keep the nitrogen on farm. So those are sort of our two big sections, but these also break down into multiple sort of subgroups. Oops. Sorry. So the various strategies that we have are all here shown in this sort of web. Jonathan and I are not working on all of these strategies, so we are going to be upfront in saying this is a very large team, this is a very large collaborative project. And so today, when I talk a little bit about the cold tolerance side of things, I'll mostly be talking about cold tolerance measurements and some of the cold germination and frost tolerance, where Jonathan will be talking more about the nitrogen remobilization project. And so there are other people doing these other sections here on this web. They are just not Jonathan or I. And so we felt it was best to just present the things that we're working on today. So the way that some of this starts is actually with some really great field level modeling. So Mitch Baum worked on this project and using AppSim. And so AppSim integrates the crops, the cropping system, soil, and weather. And so Mitch started by creating sort of a baseline, assuming that was an Iowa field. So the Midwest of the US is sort of our target environment for improving for where CERCA corn would be grown. So here he assumes that there's corn following soybeans. There's 30 years of weather data in this model. It assumes a May 1st planting with one pre-plant nitrogen application, conventional tillage practices, an appropriate relative maturity for the area, and a base growth temperature for maize of 8 degrees C, and then a grain nitrogen concentration of 1.25%. So then the idea here, or the things that we've learned from this model, is that we can look at changing the levels of grain nitrogen or changing the temperature or other values that are in the model, and then see how that impacts our overall yield. So one of the things that they've been looking at is if we decrease the amount of grain protein, do we still need the same amount of fertilizer? And the answer is actually no. So here, this model is showing you that we can use 40% less fertilizer, but only have a 0.75% of grain nitrogen concentration and still achieve the same yield levels, if not better yield than we were before. And this also ties in with cold tolerance. So here the model is comparing maize's lowest growth temperature to other species like wheat and rye. Wheat and rye are very well known to be able to grow at 0 degrees C or 1 degree C, germinate and continue to photosynthesize fine. And so here this led us to think that maybe a temperature like 4 degrees C would be possible for maize, right? So here Mitch has modeled 0 degrees, which admittedly is ambitious, right? But here you can see the yield bump that comes from increasing the cold tolerance and being able to lower that base temperature of growth. So using a base temperature closer to like what wheat has improves the yield by 22%. So then overall, what we hope you can take away from this modeling is that we can lower protein and also use that to enable lower inputs. So here, if we have, business as usual, this is what our yield map looks like. However, if we use low protein maize with the same fertilizer, we decrease 50% of the nitrogen stress and increase 10% on yield. And then in our last simulation, we have the low protein maize, but also a lowered amount of fertilizer by 38%. We can maintain that similar yield with a similar level of nitrogen stress and significantly lower inputs, saving farmers and everybody else money. So now when we think about what's actually going to be needed for the plants to be able to be grown in this expanded window CERCA-like environment, one of the first things that we come across is that to plant earlier, to capture the nitrogen, we're going to have to improve cold tolerance in maize. And so I just want to start out by acknowledging that cold is a very relative term. And then from plant to plant, from species to species, the amount of cold is relative, but also the mechanism of stress-related genes and proteins can differ. So here we're looking at some work where we're actually just testing one specific freezing response protein and looking for the presence of its product after a stress or before a stress. And so here in the chart in B, you can see we have the control amount of this stress lipid on the x-axis with the cold amount on the y-axis. And so maize is not ideal in this situation because it's actually producing more of the stress lipid just when it's sampled than in response to the actual stress. So what we're hoping for instead is to be able to achieve, patterns more like what we see in pea or Arabidopsis or in some of those, mosses and liverworts up in the upper right part of the graph there. And so we want to be able to have the plants acknowledge that it is a stress and recognize it as a cold stress, which they currently are not doing, at least at this level. And then also what's important to note is that the levels of stress are different. So chilling stress is not the same as cold acclimation, which is also not the same as freezing stress. So in maize, a chilling stress is going to be reversible. Can you pull up the first bullets, Jonathan? Thank you. So it's a suboptimal temperature, but is reversible. So in maize, we would say that a chilling stress is generally between 10 and 15 degrees Celsius. We're going to start seeing things like decreased leaf expansion, reduced stature, decreased chlorophyll content, but those are all recoverable. A severe chilling stress would be then down to about 0 degrees. Cold acclimation is a separate process. It's a period of exposure to low, but also non-freezing temperatures. Not all plants can do this because it's an additional adaptation, but what we would expect to see during this time would be the accumulation of osmolytes like sugar, salts, and proline in an attempt with the cell to push the freezing point of the water in the cell lower. And so this cold acclimation step is important for surviving freezing. And then lastly, we would have a freezing stress, which would be below 0. During freezing, ice crystallizes in the apoplast and causes an intense water potential change. So water rushes out of the cell. This leads to a dehydration stress that can actually be more intense than drought. And you can lose up to 90% of the osmotically active water in the cell. Because freezing responses are so similar to drought responses, there is a area of work that suggests that some of these have evolved from each other, sort of a chicken and an egg argument about was it the cold stress or the dehydration stress first. But these are potential pathways that we can look into tapping into and initiating in maize in hopes that we can improve the cold tolerance. So our first target is to grow below 10 degrees Celsius. So 10 is typically about the base temperature of maize right now. And so possible strategies for doing this would be to maintain our photosynthetic capacity, obviously important as you're moving into growth and trying to expand. We would want to use cold-induced promoters in these scenarios and look at introgressing genes from tolerant relatives while improving all of our membrane adaptation because the membranes are so important for surviving these stresses. To engineer that, we'd be looking at maybe, lipid metabolism, photosynthesis-related genes, and calcium signaling. This would all be in early development, looking at roots and leaves. These are some of the key pathways and molecular components. Some of these are, very well-known pathways: calcium signaling, CBF and core, transporters, those sort of things. And the biggest questions that remain in thinking about initiating lower temperature growth are knowing, first off, what are the genetic components behind the physiology of maize cold tolerance? So we know that variation in cold tolerance exists already in maize populations, and we also know that it exists in maize relatives. So trying to understand what's the difference between, our non-cold tolerant maize and those that are more prepared for low temperatures. Looking at if the same genes can confer it in maize, and then looking at relatives like miscanthus who are really great at repairing their photosynthesis proteins to allow growth to continue. Our next target is then germination at 6 degrees C. So it's important to note that any of the concepts that apply at 10 also apply at 6. These steps kind of build upon each other. So there we'd want to encourage root growth because the idea is we could keep the shoot below the soil for a little bit longer and keeping that growing point protected from any freezes. So delaying emergence and minimizing our oxidative damage. Looking at engineering would be increasing like raw scavengers. And here mainly we'd be thinking about the roots. There's actually a lot of connection between cold tolerance and root growth. Key pathways, again, are more raw scavengers, hormones that induce root growth. And then here our big questions are what strategies are already used by tolerant Poaceae to germinate below 10 degrees, which is maize's current germination temperature? And can we encourage this root growth for establishment without being detrimental to then the developing shoot as well? And so then lastly, our goal here is to make a frost tolerant maize. So that if you have emerged and had that, truly that late season unexpected freeze, survival to -2 degrees Celsius. So this is, again, the 10 degree and the 6 degree concepts, in addition to frost tolerance. Here, so we're looking more at membrane fluidity and thinking about things like ice binding proteins that help to keep the ice structure small in the cell. Genetic engineering or introgressing any of those sort of cryogenic and lipid modulating proteins in the early vegetative stage. So this step would be very like biochemically focused in, you know, those sort of pathways and components. And then lastly, wondering what sort of freezing tolerance strategies are already used in species like rye and wheat that are actively growing at 0 or 1 degrees that can improve frost tolerance in maize as well. So as a little glimpse of what I'm actually working on to help try to accomplish these goals, I just want to introduce this population of interest that we've been looking at lipid biochemistry on. So this is the B-Zea population. It's 82 teosinte accessions crossed to B73. And we have all of these BC2S3s from all different areas of teosinte backgrounds. And so when we're thinking about lipid biochemistry, we just need to know a few terms. None of this is going to get very biochemistry heavy, but we want to think about lipids as two distinct groups. There's the fatty acid tail and the head group. And the chemical properties of each of those impact how the lipid fits into the membrane and how much space it takes up. So the composition of the lipid actually ends up impacting the membrane architecture because lipids end up having different shapes. So we have some of these cone-shaped lipids and some of these rectangular-shaped lipids. And our goal is basically to balance the two so that we can keep a firm membrane without making it shatter. And then here is the evidence where the amount of double bonds in the fatty acid also impacts how much space it takes up. So we can generally classify then these lipids into fluidizing and rigidifying. And we want to balance the amounts of these during a time of stress. And that's because the lipid architecture and the phase of the membrane is impacted by the temperature. So here we're mainly wanting to keep our membranes in this liquid ordered phase. So liquid disordered, when it gets hot, then you start to have leakage. Or when it gets cold, you get phase separation and these phase fusions. So you can imagine this like tunnel-like structure in 3D coming in and out of your screen. And that's what your membranes are doing during a freeze when you have all of that water lost as they're connecting and fusing together. So then when the plant thaws, those membranes just rip apart and cause cell death. So now we think about, okay, if this is what's happening in a single cell or in a leaf in a greenhouse, right? How do we apply this bigger concept to a field or a large population like B-Zea? And so this picture is courtesy of Joe Gage's drone, is Ruben's group with several helpers from the Strabel lab and the Gage lab, sampling all of our lipids in the field. So here we have plants around V4. We manually punch each plant or each plot. And then those come back to the lab where we extract the lipids, concentrate them, and then run them on the mass spec. So we can do an untargeted lipid survey. And identify what lipids are present in all of these different accessions. Then that combined with the genetic data allows us to use these lipids just like any sort of other trait that you measured, whether it's flowering time, plant height, ear height, etc. So we check all of these compounds for things, like excessive variance. We're normalizing to internal standards. We can correct for field plot design, all of these sorts of things. And right now we're currently have screened through all of the data from the 1,500 plots that were sampled in the summer of 2024. And we're identifying like very solidly about 150 compounds, but then those need to be scanned through for excessive variance and running statistics on and that kind of thing. So, and then because metabolism, especially in lipids, is so interconnected, we're often actually not going to be looking at the value of just one lipid. So instead, we'll end up taking a lot of ratios, looking at substrate to product relationships, or looking at sums of entire branches, and maybe ratioing even some of those branches. So instead of just looking at A or B or C, Instead we would want to look at everything in orange versus everything in green. And thinking about metabolism as a bigger picture in that way can help us find more meaningful relationships that are more likely to have a large impact on these populations as opposed to one specific lipid. All right, so now I'm going to give it back to Jonathan so he can explain the nitrogen stuff.
Yep. In the beginning of the talk, I talked a bit about how the microbial processes in the soil control like this release of or nitrogen leaching. So if we take a look at this the role of biological nitrification and nitrogen leaching, we can just see how that flows goes in this slide. And so we can see that the nitrogen fertilizer contributes to the nitrate pool, which then is the one that can lead to leaching or nitrous oxide. The microorganisms nitrification processes can control this different pools. So this is the actual, like the current scenario. And this is what we want to do with CERCA, which is reduce the biological nitrification that contributes to this nitrate pool, increase residue, nitrogen, and recycling, and pools of organic nitrogen are in a way that is not easily accessible to these microorganisms that use this pools of different chemical forms of nitrogen and mineralization and nitrification processes. So one way to retain nitrogen on the farm would be to enhance the production of compounds that inhibit this biological nitrification processes called BNI metabolites. The strategies would be to activate early season root exudation of these compounds because we're going to plant earlier. If we can trigger root exudation of these compounds early in the season, we could control the amount of nitrogen that is released, and then increase the off-season biological nitrification inhibition activity via the exudation of these compounds or the accumulation of BNI compounds in stover. The way we could engineer these is just first to enhance the synthesis pathways for BNI compounds, enhance the expression of root BNI efflux proteins, and the developmental stages would be probably we're looking at B1 to B3 and around after black layer. The key tissues for this BNI are roots and stover. And the key genes and molecular components are the metabolic pathways that lead to the production of compounds that have been recently discovered to have BNI activity in maize, like CNone, MBOA, and HD-MBOA. And the unknowns and key questions about this is that we actually don't know what's the residence time of these compounds and what is the actual effective concentration that you need in soil for compounds to inhibit the nitrification activity of microorganisms. And then we also don't know what's the effect of lining and stover breakdown on nitrification. So those are some research questions that we'd like to explore. Also, I think when we were writing this paper, this perspective, one of the objectives is to engage with the plant science community and just kind of, we really like to have this section of unknowns and key questions to kind of see probably someone knows this and reaches out. So part of the objective of this paper is especially these unknowns and to highlight these unknowns and key questions to get some engagement from the community. Because this is a very ambitious project. So another of the CERCA targets is to reduce the grain protein from 8% to 4%, so cut it in half. And The target, a way to get there is to reduce insoluble protein content in the grain. So yeah, beans, the main proteins in the endosperm, and bad quality proteins, we would like to reduce their content and probably enhance the content of alternative grain soluble proteins. The engineering strategies are, they seem straightforward, but they're not knocking down or down-regulating genes, but there's a lot of gene redundancy in genes. You knock out one, there's a bunch of more genes in the maize genome. And then the expression of alternative soluble proteins, developmental stages, there's a variety of them, probably around R2, R6 when the grain is being filled. And the key tissues are the endosperm where most of the grain protein is stored, and the vedal, which is the basal endosperm transfer layer, which is a key cell type involved in the transfer of nitrogen to the grain. The key genes are, it's just one, the genes are right now our main target. And then one of the key questions is, what is the minimal zein content that can be achieved without compromising grain quality? So mutants with low zein content, like the opaque mutants, they have compromised grain characteristics, and these affects mostly their storability. So and the hardness of the grain. So you get the soft grains that are bad quality. So we want to know what is the minimal zein that we can have without compromising that. And yeah, the gene redundancy and the compensatory mechanisms could be a problem down the road in trying to knock out the genes. We also would like to recycle 50% of the plant nitrogen, and the way we want to do this is to generate alternative nitrogen sinks in the plant. One of the things could be the cob. The strategies that we have right now are activating that nitrogen sink in the cob and reduce the nitrogen transfer to the grain. The cob, we're thinking of it as a great way to have a slow release to leave a slow release nitrogen on the field. So yeah, the way we could engineer this is downregulate the nitrogen transfer to the grain and activate nitrogen storage in the cob pith. So key tissues for this remain the endosperm and the vedal, but then there's also the placenta child cell region, which is the place in the cob where this nutrient transfer takes place also. And yeah, the cob itself. The key genes and molecular components for this are amino acid permeases and synthetases, cob developmental regulators, and nitrogen and oligopeptide, amino acid and oligopeptide transporters. And yeah, on those and key questions, well, what is the cell type of specific regulation of these genes that regulate the nitrogen import into the grain? And what is the key developmental stage to change or engineer the expression of these genes? Because one of the things that we are very much aware in CERCA is that we cannot harm yield. That is like yield is untouchable. That is one of the mottos that we have. So yeah, what's the key developmental stage for this? And still in this recycle of nitrogen target, the other way we can do this is to generate a nitrogen sink in roots. We were thinking the other day, kind of like stay green leaves, we would like stay alive roots. And the way we could do this is by remobilizing nitrogen to the roots. There's two ways we can go about this. We would like to trigger root development late in the season and to trigger the remobilization of nitrogen back to the roots after grain filling has been completed. This is key because we don't want to hurt yield. So we would like to trigger this remobilization of nitrogen back to the roots after black layer. And so the key genes and molecular components for this is to find out late season underground developmental regulators and genes that are expressed after black layer in roots, which is to my knowledge, a very underexplored area of gene expression. Most of the analysis of gene expression takes place before black layer. So we now need to know what goes on in roots after black layer. And yeah, and also in key questions, what are these transcriptional regulators that could be activated in roots? And can underground developmental regulators from wild relatives of maize activate could be activated? And yeah, so when we think of reactivating root development late in the season, we logically we have to think about the perennial relatives of maize, which actually do this, particularly the temperate grasses that overwinter um, every year, and they have this ability to survive by remobilizing energy, well, nutrients, and of course energy to their roots, and hide it underground at the end of the season. So yeah, maize relatives have these traits, and they actually have this cold tolerance traits that we need to enable the early season shift. So CERCA is currently very interested in this characteristics from perennial relatives of maize, particularly Trypsicum, Cia diploperennis. And yeah, we're hoping we can get the genes uh, from those maize wild relatives. So yeah, just talking about a bit about what I'm doing, this little bit of circuit that I'm doing, all together, of course, with other Buckley Lab members like Lad, Orden, and Shankai Shu. We have been focusing on developing this gigantic survey of root RNA expression in perennial wild relatives of maize. We have been sampling like 10 to 12 species, including Trypsicum, Diplo, um, and other temperate grasses like Sorghastrum nudens. And yeah, and the way we're going about this is we're growing these plants in the fields or in pots and developing systems that enables us to track gene expression in roots. So there has been a lot of digging these plants out of the field and sampling their roots, which is a lot of work by all the Buckley Lab members. I'm very thankful about it. We were sampling like at probably 30 degrees Fahrenheit, 0 degrees Celsius in October, and just washing roots at that temperature can be a little bit of a challenge. So yeah, but the results is that we have identified some genes like the one I'm showing here that are expressed like around that time at the end of the season, and we're hoping we can transfer these genes to maize using genetic engineering um, to increase the nitrogen that's remobilized underground after this grain filling, to increase the return of nitrogen from the crop at the end of the season. The target that we have for this nitrogen remobilization is basically starting from 0%, and we'd like to take it up to whatever we can. Yeah. And yeah, this is the end of the talk. There are some take-home messages. Mostly, the objective of CERCA is to increase agriculture nitrogen efficiency, and we think we can make more efficient. Maize is already great, but we can make it better. By doing these early season shift, we think we can reduce the spring losses with cold tolerance and biological nitrification inhibition. And we think by activating this return of nitrogen at the end of the season, we can reduce the demand by recycling the nitrogen, and then by decreasing the content of low quality proteins in the maize grain, we can effectively reduce the fertilizer demand. So those would be the take-home messages for CERCA, and here are the institutions that are, yeah, providing the funding for this gigantic project. I just really would like to recapitulate what Allison mentioned. The CERCA project is a very big project. It is a collaboration between 30 public labs. industry partners. And yeah, a big thanks to all of the CERCA members that have contributed to writing this paper. Writing a paper with more than 30 people can be challenging. So yeah, thanks to everyone. And yeah, we're not putting any specific names because the list would be, it's so long. But yeah, thanks to everyone who has contributed to this perspective. And I don't know if you want to add anything, Allison.
No, just again, reiterating that this is a group effort and we're really grateful for all of the various expertise that all of the different people have. And if you have other ideas of, you know, things that we're missing, like, please let us know.
All right. Thank you, Allison and Jonathan. I see that Victor, you're here. If you want, oh, you're just asking about the PowerPoint file. Yeah, we'll be happy
I am
to.
here. I
We'll
am
be happy to
here.
share that.
I am here, and that was great stuff, guys. Great stuff.
Thank you. I think that Jonathan intends to put the PowerPoint in the CERCA General Slack later.
Yeah, I'll upload it to the Slack. Yeah.
Good luck on this very complex project.
All right. Don't see questions in the chat. Is there anyone that wants to ask any question? I'm familiar with the project, so I don't have many questions.
So, Allison, I'll ask you a question. So do you think, what do you think wheat, I mean, you listed that a lot of the inspiration for cold tolerance is from wheat and rye. Do you think there's something special that wheat and rye have that Trypsicum dactyloides is missing?
That's a good question. I don't, so my thought would be that it is very likely that these processes, these pathways are still there. They're just not being activated in the same way in some of the closer maize relatives. Particularly, I'm thinking because we know that the drought and cold response is so similar, and some of the closer maize relatives are very well known for being very drought tolerant. So I'm wondering if, or thinking that potentially these pathways that are being activated are likely still there, but have maybe been co-opted for another stress, or are just not being activated in quite the same way, and then are not as effective. That would be my thoughts. Because cold
Okay,
tolerance has evolved multiple times across, the history of evolution. So.
I do have a question, a physiological, technical question for you, Jonathan. So you were saying that you were interested on identifying genes, etc., that would be upregulated after the black layer has established. I don't know if I understood that correctly. You are not expecting to be able to remove nitrogen from the grain into the plant after the black layer has been established, right?
From the grain into the plant. No, like taking nitrogen back from the grain. No, I think the strategy right now would be to decrease the import of nitrogen into the grain. And then after black layer, nutrient transfer is like terminated. So we are looking at this window after black layer because we don't want to harm yield in any way. So yeah, I don't know if that answers your question.
So you're just using the formation of the black layer as a developmental time point to look at genes that may be upregulated in the root or in the leaves that help you to remobilize nitrogen from the leaves into the roots. But my point is that once the black layer is formed, the nitrogen that has made it to the grain is locked in. You can't remobilize that nitrogen. Is that correct?
Yeah, I think that's correct. But
No
we're
chance.
looking at what's that?
Yeah, there's
Uh,
no chance you're pulling that nitrogen back out once it's around
yeah,
the starch granules. No chance.
no, I think the black layer is actually a formation of dead tissue, and it's like kind of sealed. So I think you cannot get anything out back. Uh, yeah, we have not considered that because of that. And we are very interested in like there has to be when the black layer is formed, there has to be by a little amount of time, there has to be like a stimulus that is triggered by that. We right now think this is actually Ed's he thinks there has to be like a sucrose or a carbohydrate peak. I think there has to be a signal, probably also a peptide or something that is triggered by the physiology. You have a bunch of metabolites that were going into the grain and are not anymore. So we are really interested in that signaling pathway that gets activated after that, so we can use it to express the genes of interest that we have.
Yeah. Okay. So that's the part that I want you to clarify, Jim.
Yeah, maybe the wrong folks of the project to be asking this to, but it's more to like an agronomy question, which is if that there's an asynchrony between when the nitrogen is in the soil and available versus when it's being used. I guess why not, or why aren't farmers just waiting till the corn has emerged and then side dressing? I don't know, this is probably something I should know, but I guess I don't. I don't know how often that's done and why it's not done, if it's not done.
A lot of it is, I think, Jim, I don't think we have the right people here. My understanding is, a lot of it has to do with when the number of passes people think they can get through and timing wise in the spring and around planting time. And the over winter losses of nitrogen, it's not good, but it's not as bad as you would think it is, okay? So economically, if the nitrogen is priced low enough, with enough discount, that loss kind of gets made up. Okay, so nitrogen fertilizer in fall and winter is cheap. And so that's kind of the equipment is available and it's put down there. So
Soils are dry.
I also think some of the idea too is that you want the nitrogen in the soil before the corn is emerging, right? Because the nitrogen is so necessary for supporting photosynthesis and getting proteins going, that if you apply it pre-plant, then the minute that you start to germinate, you're having the right amount of nitrogen available.
But Jim, I think, and this is where people are doing a lot of work on, planters that inject nitrogen. I mean, a seedling is tapping into such a small proportion of the total soil volume, right?
Yeah.
This is where whether the seed's coated with nitrogen, you're injecting nitrogen right along the planting furrow, whatever, all those things are much better strategies for the first V3, V4 type of thing. And yeah, I think, and if the plant eventually needs half as much nitrogen, then actually the nitrogen infrastructure to move fertilizer is plenty large to provide the nitrogen through spring and summer. For those of you who don't know, there's a big key of how we get nitrogen fertilizer to the Midwest is there's a big pipeline that goes from the Gulf states and moves ammonia directly to the Midwest. And that pipeline gets maxed out at times of the year.
Okay, so thank you, Jonathan and Allison. We're at top of the hour. Thank you all for coming, and we'll see each other again next week. Bye,
Bye.
everyone.